Welcome to the flip lecture on genetics, specifically looking at Punnett squares, and we're going to be delving into just the simple basics of Punnett squares in this flipped video. Remember, double check with your particular syllabus, etc., to see how much depth you need to go into and when you need to stop listening or you need to continue taking notes. But I'm a big believer that learn as much as you can, because later on it's going to be a revision anyway, or it just gives you greater appreciation. Alright, so Punnett squares. Now, in genetics, when we want to determine the chances of offspring having particular features, we rely on the Punnett square genetics. We're going to do very basic ones in this particular video, but it can become more complicated. And if you're looking at a career at becoming a genetic counsellor, this is the area that you're going to get into, because couples will come to you and say, we want to have a child, we've got this particular genetic history. What are the chances of my children having this particular maybe disease or something? And you've got to tell them the chances. And so it's a fascinating area. So let's look at some basics here. First of all, let's just keep it really simple. Let's say we're starting off with brown and blue eyes, and of course we're going to use the very basic definition here that they are determined by one particular gene with two forms of an allele. Okay, so we're going to assume that B is dominant, and that's going to represent brown, right? And then lowercase is recessive, and that's going to be blue. Okay, so it's always a good thing to write these things down so you know exactly what the letters represent, and choose letters that are easily recognized and you don't confuse yourself. So we will see in some problems that you don't have to have these starting with the letter of the feature that you're talking about, right? So choose letters that are easy to remember and that is very different with how you designate dominant and recessive. So let's say we have two parents, and so one parent here is homozygous dominant, right? And so this can be the male, and we'll just write everything down. So we've got, and that male is having a child with a female who is blue eyes, homozygous recessive. Okay, so here we have the genotype, so let's write that in. I wish I had a green around somewhere. There we go. I think this is green. Yes, it is. So up here, this is the genotype. And I haven't written the phenotype, so let's write the phenotype in here. So we've got brown, and we've got blue. So remember, the phenotype is what you look like, the genotype is what you have as your gene, right? So gen for gene. Alright, so let's do this cross and we'll draw up a Punnett square. So we're going to draw this Punnett square over here. And what we'll do is, well, before we draw the Punnett square, let's just think about what this means in terms of cells, etc. Remember, in the male here, every single cell in your body, right, somatic cells, every single cell is going to have this copy of the allele here. It's going to be homozygous. However, in your gametes, your sex cells, it has half the chromosome number, okay? And so therefore, we've got to think about, whoops, sorry about your ears, we've got to think about splitting these up. So if we think that, it's a bit small, that, let's just do a big one. If we want to think that there's a B and there's a B, and they're in sperm, right? So you've got half of these being split up. And of course this is in the female here, so that gets split up during meiosis, and so in each egg, we're assuming, under normal circumstances, looks like two eyeballs. Okay, and then we have, anyway, so we have each egg, or each ova, has a little B in it. So now we've got to bring these up. What would happen if this big B combined with this big B with that one, etc.? And what we do is a Punnett square. So let's draw these out. Squeaky squeak. Alright, so we have the male over here. Let's just do the male. And then we have the female. Okay, and then we just cross. So we've got big B and little b. It's all the same for every single possibility. So the chances here is that from our offspring, you can see no matter which way it happens, we've got 100% being the same thing. In other words, we're just going to produce children. All our children are going to have the same genotype, right? And so you're going to have 100% genotype ratio, right, of capital B, little b. So 100% heterozygous. In terms of a phenotype, they're going to be 100% brown eyes. Pretty simple. Now this particular cross is called an F1 generation, right? Because we're starting off with pure breeding organisms, or pure breeding. So this is known as pure breeding. Homozygous dominant and homozygous recessive is pure breeding. So here is the first cross, and so this is the first filial generation, it's called, so F1. And what we're going to do is we're going to get two of these, obviously from two different, because we're talking about eye colour here, from two different individuals, not from the same family. But we're going to get two heterozygotes and cross them and see what we get. So I'll rub this out and we'll do that cross now. Let's do the cross. So we have capital B here, lowercase b here, capital B, lowercase. And when we do the cross here, we end up with two capital B's, so homozygous dominant. We have two lowercase b's, so homozygous recessive, and then of course we have two lots of the hybrid. So let's look at what happens here. In terms of our genotype, what have we got? Well, if we say that each square represents 25%, so this is whole thing's 100%, then we have 25% homozygous dominant. We have 50% heterozygous or hybrid, and then we have 25% homozygous recessive, which is 1 to 2 to 1. Okay, in terms of genotype. What about phenotype? Well, let's have a look at phenotype. We have one big B, big B, big B. So all of these will look the same, brown. So we have 75% brown and 25% blue, which means a 3 to 1 ratio. So this is the F2 generation. This is the filial generation, F2, second filial generation, so F2. And so in terms of genetics, what we say is a characteristic Mendelian ratio, because it was Gregor Mendel who first worked this out with pea plants, studying pea plants. He started with pure breeding plants for round seeds and wrinkled seeds, and noticed that the first generation gave 100%, just like we had, 100% of the dominant characteristic, the dominant gene. And then he got two of those in the second generation, just like us. We got two of them, and then all of a sudden a recessive characteristic came out again. So the first F1 generation tells you which characteristic is dominant. In the F2 cross, then the recessive gene comes back, and so we can see the recessive here is blue. And in our first example, the F1, all of them was brown. That was the dominant trait. And so a Mendelian ratio is a 3 to 1 in the F2 generation cross. And so here we can see F2, here's our second cross, two heterozygotes, and we get our characteristic 3 to 1 ratio. Okay, so that's Punnett squares, the basics of Punnett squares, how you do them, how you write them out, how you determine genotypic ratio and phenotypic ratio, mixed in with a little bit of Mendel. Okay, all right. We might do another video on some more crosses. Keep it simple and then work our way up to more complicated versions such as codominance, etc. Sex linkage gets really interesting. Blood groups, even more fun. But the best of all is a dihybrid cross. Okay, this is called a monohybrid cross. Mono meaning one, because we're only looking at one feature, eye colour. Anyway, I'm getting ahead of myself. See you next video.